Another thing that we often wonder about are the actual number of bacterial cells present. So while we're talking about colony morphology, we also want to talk about kind of the quantity of bacteria. So with all bacteria, they have what we call a generation time. And a generation time is how long it takes for them to double their population. So it's basically how long it takes for one cell to divide and then become two, and then for those two to divide and become four, and then those four to divide and become 8, and then 8 to 16, etc., etc. Bacterial cells do always double. So this generation time is basically how long it takes them to double, and for most bacterial cells, their generation time is somewhere in the range of 30 to 60 minutes. So assuming that they have favorable conditions with access to nutrients and moisture, they will double every 30 to 60 minutes. Some bacterial cells are much faster, so for example, E. coli typically has a generation time around 20 minutes, whereas others are much slower and can even grow with doubling rates happening every 24 hours. So it really does depend on the species of bacteria. But assuming that you know some standard information about these species, we can actually calculate how many bacterial cells will be present. That's again assuming that we know kind of three things. The three things you have to know are how many bacterial cells you're starting with, what their generation time is or their doubling time, and then also how long you're going to let them grow. And if you know those three important pieces of information, you can put them into the generation time formula. So these questions that I ask will often look like this. So this is an example of one here. If a single E. coli bacterium has a generation time of 20 minutes and is allowed to divide for 1 hour, how many cells will be present after the hour is up? So if we look at our formula is to take the total time, which for this specific example is 1 hour or 60 minutes, and divide that by the generation time, which for E. coli is 20 minutes. That is going to give you the number of generations. What that basically tells us is how many times the cells can divide. It doesn't tell us how many cells will actually be present at the end of that division, so we have to continue on. But at this point, we will know the number of generations. So we take this number, and what we do is we do 2 raised to the power of whatever we got here. And this 2 always stays, it never goes away. Bacterial cells always double, so this always needs to be a 2. But you just raise it to whatever power you got up here. And then you multiply that by the number of original cells. And that is going to give you your kind of n total number of cells. So for this specific example, we would take the total time, which is 60 minutes, divide that by 20 minutes, which is the generation time, and that gives us 3. So we know E. coli, if it takes 20 minutes to go through a doubling cycle, and we give it an hour, it's able to do this 3 times. But we want to know, after those 3 times, how many bacterial cells are going to be present. So we take 2 and we raise it to the 3rd power, and that's going to give us 8. But we're not done. We need to take 8 and multiply it by the number of original cells, which for this example was just a single cell. So our final answer would be 8 cells. Now there are some additional problems for you to review in D2L. They do have the answers worked out, so just kind of try to make sure that you can do them on your own first, and then you can cross-check to make sure that you got it correct. But to look at a few examples right now, Staphylococcus aureus has a generation time of 30 minutes, so that's our first kind of important piece of information. If we start out with 2 bacterial cells, how many will be present after 3 hours? So the first thing that we have to do is take our total time. So if we convert 3 hours to minutes, that's 180, I have to excuse my writing, 180 minutes. And we're going to divide that by the generation time, which is 30. And that is going to give us how many cycles it can actually go through, which 180 divided by 30 is going to be 6. Then we take 2, remember always 2, it never changes because bacterial cells always double, and we raise it to the power of 6, which is what we got here, and that is going to give us 64. And then the last thing we need to do is take 64 and multiply that by the number of original cells, which was just 2, and that's going to give us 128 cells, which is our final answer. And then for this one, we look at Streptococcus pneumonia has a generation time of 5 minutes. If we start out with 5 cells, how many will be present after 25 minutes? So we need to take the total time, which is 25 minutes, and we need to divide that by the generation time, which is 5 minutes. And that is going to give us 5. So then we do 2 to the 5th power. That is going to give us 32. And then we need to take 32 and multiply that by the number of original cells, which was 5. And that is going to give us our final answer, which is 160. So while these problems are meant to kind of show you the math portion of it, I do want you to understand these numbers are not really realistic. Certainly when we inoculate a plate, we're definitely adding more than, 1, 2, or even 5 cells, and we're certainly going to get numbers higher than 160, 128. So just keep in mind, these numbers are definitely not an accurate depiction. The numbers that we're typically working with are usually in the millions.